Hello everyone, I am Suraj Pochampally and I'm a graduate research assistant in the Department of Mechanical Engineering here at UNLV. Today I'm going to present a poster titled Production and Characterization of Physical and Chemical Modified Walnut Biochars for TC and PC Adsorption. This project was funded by Savannah River National Laboratory and we're very thankful to them. Biochar is a biomass-derived black carbon that is recognized material for soil amendment, soil improvement, and has also been used for water treatment. It is produced from waste biomass feedstocks like walnut shells, peanut shells, wood chips, etc., using a process called pyrolysis, where it is heated to a very high temperature under the absence of oxygen to produce a porous carbon material. Trichloroethylene, which is TC, and perchloroethylene, which is TC, are carcinogenic contaminants found in groundwater and have a maximum contaminant level of 5 micrograms per liter. But at SRNL, the maximum TC concentration was found to be in the range of 10 to 10,000 micrograms per liter, so they need to be treated. In the study, we gathered big-sized walnut shells and we crushed and sieved them into small-sized walnut particles that have a size of less than 850 microns. Modification of a biochar can be done either before pyrolysis or after pyrolysis. Figure 2 shows the after post-pyrolysis modification techniques. Figure 3 shows the pre-pyrolysis modification techniques. In this study, we found out that pre-pyrolysis modification is a better technique because it is energy efficient and it produces a better performing biochar. So we decided to focus on the pre-pyrolysis modification using three different methods. First is an acid modification using hydrochloric acid or HCl. Second is a base modification using potassium hydroxide or KOH. Third is a steam modification using DI water. For each modification method, we soaked the biochars in these liquids for 4 different soaking times: 3 hours, 6 hours, 12 hours, and 24 hours, and then characterized them using 2 chemical and 2 physical techniques. The first characterization is hydrophilicity test or water contact angle. As you can see here in figure 4, if the contact angle is greater than 90 degrees, the biochar is hydrophobic. And if it is less than 90 degrees, it is hydrophilic. We measured the contact angles for all the biochars and we found that for acid modified biochar, as the soaking time increases, the hydrophilic contact angle decreases, which means the hydrophilicity increases, and which is the opposite for base and steam. For base and steam, as the soaking time increases, the contact angle increases or the hydrophilicity decreases. We performed another analysis to support this, which is called FDR Analysis. In this analysis, we aim to identify the surface functional groups of the biochar, and we found out that for acid modified biochar, as the soaking time increases, the hydrophobic functional group decreases, which goes hand in hand with the contact angle measurements. So similar results for base and steam were also found. Then we gathered the images using scanning electron microscopy of the biochar surface and the pore structure. Here, the SEM images for base modified biochars are shown in figure 9, and the 3-hour, 6-hour, and 12-hour biochar images can be seen. We identified that as the soaking time increased from 3 to 12, there were a lot of small-sized particles or small-sized pores that were developed in the biochar. It can be seen here in figure C1. This explained that as soaking time increased, the biochar developed new pores which can increase the surface area of the biochar. Surface area of the biochar is an important factor because if the surface area is higher, there is more area for the biochar to facilitate the contact between the contaminant and the biochar. We performed another analysis to identify the surface area or evaluate the surface area of the biochar, which is called BET analysis. As you can see here in figure 11, the green column indicates the microporous surface area of each biochar at different soaking time. This is shown for the base modified biochars, and it shows that from 3-hour base modification to 12-hour base modification, the microporous area almost tripled, which indicates that as soaking time increases, there are more micropores being produced, which is beneficial for the biochar. In the future, we plan to optimize this modification technique to find the optimum modification time for each of the method, acid, base, and steam, and then perform adsorption experiments on these biochars to identify the best modification procedure and the best performing biochar. Thank you very much.